Hello students, so welcome to the course of biomolecular spectroscopy, where we are discussing circular dichroism. And in circular dichroism, in the last class we discussed how we can use circular dichroism for nucleic acid analysis, right. So, that is what was the topic. We looked at there are specific signature in B type, A type or G type of DNA that understand. So, can we use CDS spectroscopy to study the base stacking orientation in the DNA? That is what we let us discuss in this class today. So, from your biochemistry book, we know that in DNA there is a stacking orientation. Now, in any double helix, double stranded DNA or RNA, these bases are stacked on top of each other. They are basically, if you look at this is a helix axis, base is stacked like this, right. So, either parallelly or twisted or tilted or any of these orientation that are there. So, roughly perpendicular to the helix axis, our helix is going like this, base are stacked like this, right. So, they are stacked and they are planar hydrophobic aromatic ring that stacks, and the distance between the hydrogen bond donor and acceptor is about 3.5 or 3.4 angstrom, and therefore, they form the hydrogen bond, right. So, for formation of hydrogen bond you require this distance to happen, and generally what happens, because now it is a bulky group, right, base is a bulky group. To accommodate each other and minimize the van der Waal interaction, right, or sorry, maximize the van der Waal interaction, minimize the steric clash, they have to be adjusted something like this. So, tilt can happen, and that is what happens, this tilt helps in adjusting it. And in GC we know already that it is a 3 hydrogen bond are happening. So, base pairing interaction also gets another stability in GC compared to AT. So, these are possible base stacking, right, different stacking orientation that you can think of in the double stranded nucleic acid. So, some of those that you can look at detail. So, shear parameter, you have along long axis, stretch, short axis. So, this pulls apart, pushes together, and the changing separation that happens, stagger, buckle, propeller, opening, all these are base stacking that are there, the tip or helical twist, and they have impact on a structure. Suppose this is stretch which is along short axis and it pulls apart or pushes together, something like this, it directly determines the hydrogen bond. Similarly, this sliding, which actually it is a key determinant for major and minor groove width in A DNA or B DNA. This is again because of how base is stacked. And now, rise gives you pitch of the helix, how much 3.4 angstrom in B DNA, that is the coming in the pitch of helix because of this rise base pairing interaction. Or basically helical twist determines the number of base pair in per turn, like a 10 base pair per turn in B DNA. So, these kind of interactions are basically optimized to determine the structure, the major group, minor group structure, the hydrogen bond length, and so on and so forth. So, base stacking is important structural parameter. So, perpendicular arrangement, as we discussed, base pairs are arranged nearly perpendicular to the axis of DNA. It allows to base to stack effectively, how they are stacking, right. It can have a planar structure. So, each base pair is relatively flat, right. So, flat, and that facilitates the close contact. If they are flat, it can form the very nice hydrogen bond. And interplanar distance, that is a stack, like distance between a stacked base, one base here, one base there, that is about 3.4 angstrom. The twist and tilt, adjacent base are not directly on top of each other, you know, this is not favorable. So, they have to twist or tilt you know why? Because finally it is a if you look at, because of the double bond, some negative charge is inside the ring and some positive charge on the outside. So, in pi stacking, that we know it can be best optimized, either it is tilted or twisted or slightly adjusted, and this typical twist is about minus 30 degree. In all the stabilizing interaction, this hydrophobic or van der Waals forces basically determines what kind of stability they get. So, it is a interaction between pi orbital of aromatic ring in the base, it is a crucial for stability, right. So, this provides additional stability of DNA other than the hydrogen bond. And then there is a sequence dependence, stability and geometry of stacking such as AT versus GC depends upon base sequence, where and how they are adjusted. So, now CD, how CD can be used in this base stacking orientation. So, as we know that CD depends upon like a very sensitive tool, depending upon what is the chirality and how base stacking orients. So, in the nucleic acid, that is what changes, like as we saw, and therefore, that can be detected using CD. So, because of this base stacking interaction between the adjacent base, and then we have a chirality in the backbone, it generates a specific CD signal. So, this CD techniques relies on the fact that when the bases are stacked, they interact with circularly polarized light differently on the helical twist, inclination and polarity, and that basically determines the base stacking and orientation. So, what happens? One can use this differential absorption signal by this base stacking to distinguish the helix type B versus A versus Z. So, you know these are right handed, B positive band we see at 275 to 280, negative at 245. A or RNA gives positive signal at 260 to 270 and negative to 210, but left handed gDNA gives a negative signal at 290 and positive signal at 260, right. So, between even A, B and G, because of differential base stacking, you have a different pattern, and that is what one can use for distinguishing the A type, B type, G type DNA. Other than these double stranded the DNA, you can have a quadruplex topology. So, quadruplex means 4 strands, 2 and then 2. So, CD actually can be instrumental in distinguishing the G-quadraplex. A parallel G4 features the positive band at 260 and negative at 240, right, parallel G, right, if all strands are in the same direction. The anti-parallel G4 exhibits negative band at 260 and positive band at 295, right, and base stacking. So, these are for parallel and anti-parallel. So, if we suppose we look at these signatures, we know that in quadruplex, whether it is parallel or anti-parallel arrangement, just looking at this, right. Now, what happens of base stacking in G rich sequence? The CD reveals that certain G sequence like a DG4 from parallel stranded structure is characterized by same 260 that is found in aDNA, right. So, this is like this is a common features that you can have between A and quadruplex. Now, one can monitor the conformational change using CD. So, CD can track the conformational change induced by temperature, or if you change the ion or ligand, how it is going from B to A, B to G. We also saw this in last class, and you can also see the transition that is happening from poly DAT to pseudo Z or X DNA or various specific conformation that are populated in like a little non-natural condition like a high salt or like alcoholic condition, how your DNA is changing, right. And you can also have a local stacking via analog. So, if we replace the purine with 2 aminopurine and pyrocytosine, we can also see how these like analogs can change the specific structure by changing the base stacking. So, these all structural perturbation that may happen because of the solvent effect, because of the high salt, because of an analog incorporation can be studied using CD. So, this is an example of G-quadraplex DNA, right. So, quadruplex, you see these are parallel 1, 2, 3, 4, 4 stranded in the same direction, and basically the hydrogen bonding is like here. So, you can see 2 hydrogen bond here, 2 hydrogen bond here, and 2 here, 2 here. So, that is the hydrogen bond that is happening, and this is anti-parallel T, you can see T quadruplex, and it is a tetrad or quadruplex we call it. So, if we look at these are two basic type of guanine quadruplex that one can have, and spectra of quadruplex you can see 260 positive peak here, and anti-parallel have a negative peak at 260 here. So, whether it is parallel or anti-parallel you can find it out, and also the transition that may happen. So, transition how it can happen, because of you change the salt or the solvent condition it can have. So, quadruplex display additional characteristic positive peak at 210, right. So, here is a 210 you can see in both of the cases that is there. So, different CD of parallel anti-parallel quadruplex can originate from different base stacking arrangement. One can see here this is just one layer, but one layer, 2 layer, 3 layer. So, all these are by base stacking that is happening in quadruplex, and that can basically change the signal that is there by just base stacking. So, G-quadraplex DNA one can have an intramolecular G-quadraplex DNA that forms the slowly. So, also transition happens between different quadruplex type that are slow. Now, quadruplex formation like a say here G tetrad or T tetrad or all of these formation can happen by this metal ion. You require a metal ion which stabilizes, suppose namely potassium, although ethanol can also be inducer in quadruplex generation. So, the CD spectrum parallel to the quadruplex is similar to CD spectrum in the A form, because 260 positive that we already see is also characteristic peak in the double stranded DNA, and this may suggest that the quadruplex and A will be having similar base stacking, right. But quadruplex has also positive band at 210, you can see here that is a 210, that is where A form like a here at 210, so a negative one. So, these are some of the similarity and some dissimilarity between A form of DNA and quadruplex, and here you can have a time dependent parallel stranded of quadruplex stabilized by potassium, here sodium induced quadruplex that you can have. So, the parallel or anti-parallel you can find it out, how parallel is similar to A type of double stranded DNA you can find it out. So, these structural transition you can really find it out, and in quadruplex formation the presence of metal ion is extremely critical, those stabilizes the structure, right. So, the distinct CD signal for parallel, anti-parallel or mixed type of topology that we have. So, CD represents the highly active area of research, how the quadruplex formation happens, where how the transition happens, and you know where quadruplex is found? It is found in the human telomere region. So, you can see this is also an indication of aging. So, if you can isolate this DNA, understand the conformation, you can comment about aging or age of a person, how it is aging. So, lots of interesting research is happening on telomere region connecting with the human health and aging, and this is the CD spectrum of telomere G quadruplex conformation. CD shows that human telomere G quadruplex distinct signature for the anti-parallel, right, anti-parallel this one, and a hybrid one is a hybrid form is red, and then you have a black one which is the parallel propeller here acetonitrile. So, these kind of quadruplex structure that you can find it out. So, anti-parallel G4 like G tetrad stabilized by potassium ion that display a positive at 295, negative at 260, right, just now we saw it here and mixed or hybrid topology show feature of those. So, one can have here both features. So, CD is often first and most rapid method to find it out, confirm whether the G tetrad is or G quadruplex is present among different topological arrangement or not. Now, another quadruplex which is C, right, made up of C. So, DNA strand rich in cytosine can generate the C quadruplexes, right, and these consist of two parallel homoduplex here, right, two parallel homoduplex, and then you have the protonated ones, because forming 3 bond you require one protonated to be happen, and that is what happens. So, two duplex mutually intercalate and forms an anti-parallel orientation, right. Here you can see these are anti-parallel orientation that is happening, and that is what it is called intercalated or I tetraplexes. The C quadruplex, it is a intercalated quadruplex. So, formation of like a cytosine quadruplex promoted by this slightly acidic pH, because you see here this has to be protonated, and this protonation can only happen if the pH is lower, and therefore, it is denoted by Cc plus, the hemiprotonation state, right. So, these kind of lower pH, lower than 7 you require. Interestingly, the sequence that are prone to fold into cytosine quadruplex binds to proton at this structure even at slightly higher pH, right. So, this was similar to guanine. This intermolecular cytosine or quadruplex formed by the slow kinetics. You can see as you change the pH, it is a slowly transitioning. So, at a lower pH you have the characteristic structure of intercalated, and then as you increase like you see the structural transition is happening of the intercalated quadruplex. Now, DNA fragment rich in G and A, how you can find it out if your DNA is rich in G and A. So, you know, rich in guanine and adenine exhibits cooperative in the melting, that this is our, say suppose our sequence is GA, 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 3 prime to 5 prime, 5 prime to 3 prime. So, how one can find it out whether this is rich in that? One, of course, you can look at the CD signal and you find it out what kind of structure it populates. So, molecular structure has not been solved in spite of the several attempts that has been done, but CD has given some idea. The first conformation of anti-parallel homoduplex containing GA, the CD spectrum is like this. You see a CD spectrum like this, right. So, here 0 zinc and protonation, and the lower concentration of zinc cation induces various conformation, right. It is not two state nature. So, you can just see lots of conformation depending upon zinc, whatever it is there, it populates. So, anti-parallel homoduplex is probably part of zinc stabilized DGA and GA and TC triplex that we have, right. So, you can see various conformation that are there even in NaCl, how with change of the concentration they are changing the conformation, various kind of conformation they adopt. So, spectral similarity indicates the presence of guanine base stacking happening, and adenine probably extrude from the double helix, and those are different conformation that are taken by these. Detailed structure is not known, but spectral signature in the CD known. Similarly, you can have the single stranded here GA. So, third conformation order in the single stranded in a protonated state of adenine, and one can see if you have a lower pH, this is the structure like a 6.9, it is a neutral pH, and as you lower the conformation is changing, right. So, this indicates that guanine stacking does not change significantly in the duplex formation, and and with the pH it changes the conformation as we go from neutral to lower pH. Similarly, you can find it out DNA fragment which is rich in AT, you know AT can have a different signature than GC. So, this is the poly D 2AT. This undergoes salt induced conformational change. One can see the conformational change that is happening here, magnesium or sodium and what kind of ellipticity change that happens. So, basically you can use CD signal to monitor all the structural transition that may be happening, and therefore, now since we understand different conformation, one can use the CD to detect the polymorphism that is present in any DNA sequence. So, it was shown that DNA structure observed in the crystal, like you can crystallize it, crystallization is required for solving the structure, and when you crystallize you can see that different kind of conformation is adopted by ethanol or TFE. Now, these observations are made that DNA duplex and also guanine quadruplex are present. So, CD has contributed significantly to demonstrate the polymorphism that may happen in human telomerase DNA structure, right. So, one can see here whether it is parallel or anti-parallel or mixed parallel, all these will have a different signature, but in telomere region it can be coming from all of these structure, and therefore, you can find it out what kind of polymorphism these guys has. Similarly, lots of research is going on aptamer for detection, right. So, detection. So, and those are like this aptamer based detection is becoming popular. So, you see the aptamer 8, right. So, aptamer dopamine aptamer alone, and then you can have dopamine aptamer plus dopamine, you can see the structural transition that is happening, right. So, wavelength wise shift is happening. So, the absorption shifting, spectra is shifting. So, aptamer basically are single stranded nucleic acid DNA or RNA that can bind to a specific target, and that is why I was saying it is used for detection and often adopts this unique 3-dimensional structure upon target recognition. So, in this case target is suppose dopamine, this aptamer adopts this certain conformation. So, this characterizing the folding and ligand binding by aptamer, CD can be used, and it gives a interesting signature because unique signature of CD aptamer functional structure can be also used as a benchmarking, and that conformational change happens. CD aptamer you can see used for monitoring. So, serotonin aptamer alone and serotonin aptamer plus serotonin, when it binds to serotonin you can have this kind of a structural change. So, that is what is important, that you make an aptamer and you can show whether your aptamer has bound to a specific ligand that you have designed or not by just looking at different signature that one can have. Here is the maxima as at 260 and here you have a maxima at 290 or something like that, and once upon it like this is alone, and when it binds its signal goes up, it shifted also toward the higher wavelength. So, one can conclude here in this case that guanine rich DNA molecule are known to self assemble, and that is what it forms the multi-stranded arrangement, right, multi-stranded arrangement which is useful for supramolecular chemistry and nanotechnology. You can make this quadruplex like a structure or aptamer quadruplex like a structure for supramolecular chemistry. It is used for detection, though it is found in telomere, but one can synthesize and use for various nanotechnological application because they adopt a particular shape and size. So, it is used in supramolecular chemistry and nanotechnology. Now, the combination of DNA sequence that can isomerize between distinct arrangement represents one of the most promising nanotechnology material. Now, this DNA is used as a nanotechnology material, and once you make this you need to characterize it, and here is a CD. CD can distinguish the order condensation from chaotic aggregation, whether we have formed a distinct structure or not. CD comes to rescue. It gives you a characteristic signal if it has formed. Now, this if it is scattered, what happens? The aggregated molecule will scatter the light, and that you can use it for understanding whether it is formed a structure or its aggregates. Now, conformation of DNA in aqueous solution and crystal can differ, and CD can contribute to elucidating the relationship between DNA conformation in various environment. I saw you for alcohol environment, I saw when salt is present, when acid is changing, how CD can be used. Now, so CD is one of the most suitable method to study the quadruplex formation in various DNA motifs that we have. CD can be used for understanding this telomeric DNA, which is quadruplex, and it can be related to like a senescence on cancer, because this gives the idea about health. So, thus telomeric G-quadraplex is an attractive target for cancer therapy. People look at this presence of G-quadraplex and that can be targets, and now we can synthesize this G-quadraplex that can be used for ligand screening before we really go to cell experiment or the even preclinical stage. So, one can do in vitro drug screening or potential cancer therapeutics by taking this quadruplex. Now, understanding of this quadruplex structure and ability to monitor the formation or dissociation of this structure is important for rational drug design. So, if we understand if the molecule is perturbing the quadruplex structure, we know that this is a potent molecule. So, what we can say, the CD contributed significantly for demonstrating the polymorphism of human telomere structure, the presence of G-quadraplex, and all those CD is very useful. So, CD was one of the first method that shows that parallel quadruplex of human telomere DNA fragment that was observed in crystal and not formed in vitro. And CD can be employed to demonstrate the topology of quadruplex. Folding depends upon what number of telomeres that we have. This is called so called 3+1 quadruplex structure. And later on subsequently with a detailed analysis was done by solution state NMR for formation of this quadruplex. You can go and if you are interested, read about G tetrad, C tetrad, A tetrad that was used for solving for understanding the detailed structure by solution state NMR. But before we went into really detailed structure, CD can give you spectral signature. So, CD also provides the suggestion for long telomeric sequence or the structures on bead like a string that is present in nucleosome, and this concept may contribute to the expansion of gradual shortening of the telomeric DNA, right. So, as I was saying, this is an indication of aging and you can find it out this signature by just looking at CD. So, generally it is accepted, not yet confirmed by any other method, that shortening that can happen and can be studied, right. So, let us close it here for today. I hope we discussed about the how we can use the CD for protein and protein aggregation, protein like a, we also looked at the how we can understand the folding pathway using CD. Now, we looked at how we can use CD for DNA structure, the drug screening, drug binding and so forth. So, with this comprehensive overview of CD, let us move to the next lecture to understand the use of CD in some specific purpose in disease pathology and drug design. Thank you very much. I look forward to have you in the next class.